atypical careers avenues after STEM PhD. Currently, the perception is that you do a PhD in STEM subjects. The main avenue is, firstly, you will go into research. Otherwise, you'll go into teaching. Otherwise, in some cases, let's say if you're a chemistry graduate or a biology graduate, pharma industry is an option. But we all know that all of these fields are quite saturated, and there are a lot of atypical career avenues which are coming up, which are related to science. So we like to design sessions, call people who are experts in such careers, and they get to talk to our participants so that our participants are aware of all of these avenues. And today's webinar is very much a part of this focus, right? We want to talk about data driven and biocuration related roles, which could be a sustainable career option for life sciences PhDs, right? And along with this, along with making people aware about all of these careers, we also provide some kind of mentorship guidance and interview CV guidance, internships for motivated participants. And academia readiness is, of course, a part of this program, because we know that almost half of the PhDs are going to go ahead and teach. They are going to go into research. So we do help or design sessions in proposal and grant writing, as well as in pedagogies. So how science is taught in the college and in the school is changing now. So we design sessions so that our PhDs, when they start teaching in colleges or in schools or wherever, they are equipped with active learning strategies and novel assessment strategies, which will make their teaching learning process more engaging. Right. So these are the four focus points of the ECR program. And the program structure is that we have, such like the webinar today, some short 1-day workshops or webinars on various small themes. Along with that, we have level 1 workshops, which are 3-day on-site workshops. That means those are not conducted in IISER Pune. We go to different universities and colleges all over India and conduct these level 1 workshops. Motivated participants from level 1 workshops are invited to attend a 10-day residential workshop at IISER Pune, which is like a boot camp. It covers all of these themes and so much more. And even from these, motivated participants from the level 2 cohort will be given some industry opportunities. There will be hand holding and mentoring on a deeper level if they wish to engage in industry or other opportunities related to their or which will help them start their careers, right? So this is the program structure. And so far, so if you talk about what we have done so far, we've done 20 different level 1 workshops. We've reached more than 1,300 participants through these workshops. We've conducted 2 level 2 workshops at IISER Pune. And so the network is slowly being built, but we do need to do these smaller 1-day workshops or webinars so that we keep on engaging with the participants, and we get to know from the participants as well that what they would like us to do. So it's a two-way process, as it normally is with any kind of projects. So our resource persons for today, so with that, it was a very sort of organic process for me to think about this topic as my first webinar topic in the iRISE-ECR program. So I come from a biology background. So do most of our team members right now. So I know that for me it was a very obvious choice that after BSc I have to do MSc. After MSc I have to do PhD. During PhD I realized that I really like teaching. I love teaching, so I did not explore any other option other than teaching. But when I thought about it, that what if I would have wanted to explore something else, which is not research or not teaching, I realize that I am quite ill-equipped to, get this information or to figure out how to go about this. So the next best thing that I could think of is, okay, then what are my other friends doing? So that is where I reached out to two of my very good friends, Yutika Dikshit and Apurva Kulkarni. So Yutika has done her BSc in microbiology and MSc in health sciences. After that, she chose to work in the healthcare sector, but in a very different way. not as a researcher or not as a teacher, but she joined Merck as an associate project manager. Then she joined the company called as Evolvus, where she joined initially as a curator, so biocurator, which she's going to talk about in this webinar today. Then her graph went on increasing. She became the team leader, project manager, senior project manager, eventually a director, and a senior director data strategy. So I have seen Yutika, grow in these roles, and I have seen her enjoy her work like anything. So that is what was the point that okay, I thought that Yutika should talk to people about how to enter this career and how to thrive in this career, how to succeed, and what could be the career path in this particular, biocuration domain. And I really enjoy talking to her about all of this because like most of us, I had no idea what biocuration is. So when I talked to her about it, I understood more about it, and I thought she is the right person to talk about this domain to the life sciences or STEM PhDs who really want to explore newer careers. Similarly, Apurva is also a very good friend of mine. I met her in when she was working as a junior research fellow in ICER. So she has done her BSc in biotechnology and MSc from Vellore Institute of Technology, and then she worked in ICER Pune as a junior research fellow. I distinctly remember she was at that point. She was on the path of sort of, okay, I'm a JRF. I want to eventually start my PhD, and I'm going to do research. But I could also see that she wanted to do something else. And that's when she branched out and joined Megavision Life Sciences and became a genomic scientist. And she enjoyed that process a lot. I think she was that there, she had to do a lot of wet lab work. Then she joined Velsera, where, like Yutika, her journey was quite quite fast and quite successful. So she's also joined as a biocurator, and now she's a product manager. And I suggest, I would recommend all of you to please check their LinkedIn profiles. And what Apurva had written on her LinkedIn profile was, I thrive at the intersection of, healthcare or other science, product, and strategy. So again, here is another person who really enjoys innovating in the field that they are in and enjoys their work. So I thought two people like this would be the perfect combination for this webinar, where they can talk about how one can what is biocuration, what are data driven roles in life sciences, and how can one explore this domain. So this was my brief introduction for the day. Now after this, Yutika will first talk about biocuration, then Apurva will give us a holistic overview of the field and talk about the opportunities. After both of their brief talks, we'll give up time for some question and answers, and later on we'll have a longer time or a longer duration for question and answers. Please keep your microphone on mute throughout the session and keep your video off unless we request you to talk, or unmute. And yeah, we all know all of this. We are quite well versed with doing stuff online. So over to you, Yutika.
Thank you, Manwa. Thank you for the introduction and welcome everyone to this webinar. It is lovely to see so many people having joined in today from all over India. And that actually makes me feel very happy that I can communicate and I can tell people about biocuration today to a larger expanse. So just give me a minute while I share my screen. Yeah. Is it visible to everybody? Manwa, is it okay? Okay, thanks. All right. So before I get into what is biocuration, I would like to take a little poll from for all of you. Before today, how familiar were you with the term of biocuration? Since when you people were joining in, I saw you were coming from all types of fields, maybe biology, chemistry. So how familiar are you? Have you never heard of it? Are you somewhat familiar? I think Manwa has already shared the QR for this, or you can, as she has mentioned, go to
Yeah.
go to the other instructions and just give in a little poll so that I get an idea of where all of you stand in terms of knowing what is biocuration. Okay. Now, before I start with biocuration, I am going to first explain a little bit about a very simple concept, which is what is wet lab and what is dry lab. Okay, you might all already know this, but I thought before I begin this, I should give like a little bit of a primary concept. Wet
Yutika, just you're not I mean, the presentation is not full screen. If you could just do that.
lab. Okay,
Sorry to interrupt. Yeah.
sure, sure, sure.
Yeah.
Yeah,
Yes. Thank you.
better. Yeah.
Yeah.
Yes. So, what I want to explain before I get into biocuration is what is the wet lab and what is the dry lab. Okay. The reason I want to explain this is in case any of you do not know it, the difference is that wet lab is when you are taking a hands-on experiment using, chemicals, working in a laboratory, getting results and analyzing those results. All right. Now, a dry lab is where you're doing a computer-based research. So you do not have to attend, you do not have to go into a laboratory. You can sit at home, you can do analysis of data, you can sit at your desk in office and use computers and software databases for this. Okay, now biocuration lies in the dry lab. All right, so it is a part of the dry lab section. Now, what is biocuration? All right, it is the collecting of biological data from scientific literature and experiments, organizing it, annotating it, validating it, and then integrating it into a structured biological database. Okay, so it is basically turning raw biological data into a reliable, usable knowledge. Now, I'm going to give you a very simple example for this because all the earlier slide might sound a little confusing. So imagine a recipe app, okay? A recipe app and a biocuration app, okay? A recipe app is going to take various resources, is going to use various resources like a cookbook, food blogs, things from Instagram, things from YouTube, and it's going to create a list or a combination of recipes in a recipe app. Similarly, a biocuration app is going to use different types of journals, different types of patents, different types of publications, which are going on all over the place. All this material is used and collated into one place. All right. Now, a recipe app, if you have ever accessed, I'm sure all of you have sometime checked a recipe, they will always give you a list of ingredients. Okay. A list of ingredients that are required, how much time is it going to take, which ingredient is required in what quantity. Okay. So similarly, a biocuration app will be collecting, annotating biological information like genes, diseases, pathways. So it's taking dispersed data and converting it into actionable knowledge. Okay. So whether you're preparing a meal or you're trying to advance a biological research, the approach is where biocuration comes in, where you're trying to collect information and put it into a structured format. All right. Now, I'd like to take up one more poll right now. When doing a project, when you have, I'm sure you people have worked on several projects, what do you rely more on? All right, have you done research papers? Have you gone through biological databases? Both, or none of the above, or you're not really sure. So please go ahead and give a little poll out here so that we also realize how much of data have you accessed in the past. All right. Now, why does biocuration matter? All right. Biocuration matters because while we speak, there is more and more biological data which is being generated even at this minute. All right. So almost most of this data is fragmented, sometimes inconsistent, or sometimes even unclear. So you might feel that you are sitting here today and writing a certain amount of work, but the person sitting next to you is doing something else in a different format. All right. So biocuration is going to turn this scattered, inconsistent data into reliable, high quality data, which is going to help you with enabling new discoveries through data reuse. All right. Now, the word data reuse is very important, okay, because this data is actually already present out there. What biocuration helps you do and why it matters is because it is going to help you reuse that same data and get an actionable insight out of it. Okay. Now, I'm going to give you an example of breast cancer. Sometimes we want to look up something for breast cancer. We will enter the terminology breast cancer. and we will get a little amount of data on it. Now, I'm just going to give you a very simple example of how complex this data can be. All right. So if you just enter breast cancer, you're going to get several things. All right. Now, a breast cancer can be termed as ductal carcinoma. It can be termed as invasive carcinoma, or it can be termed as various aspects. Sometimes breast cancer is also recognized based on its receptors. So it can be a triple negative breast cancer and HER2 positive breast cancer. So this is how many data points there are to breast cancer. All right. Now, apart from just the name of the cancer, the treatment is further bifurcated. All right. If you take just chemotherapy from table 2, it is going to have tons of cytotoxic drugs. If you take targeted therapy, it is going to have lots of targeted therapies. If you want to just take a background check on, the kind of patients that have had breast cancer in clinical trials, there is a lot of family history, a lot of hormonal exposure, a lot of age related risks, which are all data points. Okay. When we talk about several data points, that is how much deeper the information is going. And that is why biocuration matters. That is why it is important to curate this information and put it in a standardized format. All right. Now, what are biocurators actually supposed to do? So, what is the role of a biocurator? So, you might feel that, what am I supposed to do if I want to become a biocurator? All right. The first and most important part is you should be able to read and interpret scientific literature. Okay. Extract key biological facts like genes, proteins, phenotypes, pathways, etc. Now, once you have extracted this information, it has to be standardized. All right. It has to be put into control vocabularies. I can give you a very simple example. The spellings is something which needs to be standardized at a very large scale. UK spellings and US spellings are always different. So even if you take up something like hemoglobin, it will be HAEMO versus HEMOGLOBIN. All right. A lot of you must have faced this problem even with your names. Okay, I face it on a daily basis. My name is YUTHIKA. People will write it as UTIKA. All right. So applying control vocabularies and ontologies to biological data becomes one of the biocurator's main role, where he standardizes the data which he has extracted. All right, then he has to validate it and cross reference it. And once that is done, he needs to update and maintain the database. So today, if you have a drug database that you're updating and your drug has gone into a clinical trial in the market and it is showing a side effect after 3 months, the data which has come 3 months later needs to be updated into that database. Okay, because whoever is going to check your database is going to see a drug going into a particular clinical trial and then see nothing. Okay, unless you update this database, which is also one of the main roles of a biocurator, then the database will not stay relevant. All right. So these are one of the few of the roles which biocurators are supposed to do. Now, have you ever come across, again, this is a poll, a terminology confusion? All right. There are lots of genes which are described differently. For example, EGFR can also be written as HER1, can also be written as ERBB1. So have you ever come across something like this? Please put in your poll for this so that we also know to what depth have you come across this confusion. Okay. Now, the types of data curated. What are the types of data curated? Actually, sky is the limit, to be very frank. But depending on which company you apply to, there are various companies, various research institutes which will be dealing in different types of data. All right. So some will be dealing in gene and protein functions. Some will be dealing in just disease associations. So it is going to basically depend on which place you are applying to and what is your interest. But otherwise, literally for curation, sky is the limit. All right. I have just put out a few major curation databases, biocuration databases, which you can at your own time go through just to familiarize yourself with what is the type of data that you can encounter when you ever take up a job or when you go for an interview, or if you want to prep yourself, for a biocuration role. If you want to just go through these databases, it will give you a brief idea of the type of annotations that are done, the type of vocabularies that are used. It will definitely increase your information and help you prep. So do go through these databases. Now, this is something that I wanted to explain because this is something that you will also encounter when you join a company or when you join any research institute. Is the place you're joining doing a manual curation, or is it doing an automated curation, or is it using a hybrid approach? When you do a manual curation, obviously accuracy is very high because it is going through a human brain, but at the same time there is a lot of time consumption. All right. Now, if you're using an automated curation, it is exactly the opposite. All right. You will have a fast and scalable process, but it might require human validation. So most of the companies will use a hybrid approach where a little bit of the basic information is automated, and then important information like, for example, gene and disease linkage is done manually. All right. So just wanted to give you an idea of the fact that you will come across automated processes also when you go into biocuration. All right. Skills required and that are needed for biocuration are basically you need a strong background in molecular biology. You need scientific literature analysis, data annotation, familiarity with databases, and attention to detail. Now, data annotation and organization is generally taught in a company. They will give you their process of doing it, but a strong background in molecular biology definitely helps as a skill. And attention to detail is a very important point here, because if you miss information, if you miss a detail, you will lose that little edge you have over an automated process. Okay. So when something comes to you from an automated process, it is important, and it is the human eye which needs to catch it at that time. So please remember this one point where attention to detail becomes one of the main factors in biocuration. And a bonus is all obviously if you already know any type of programming, or you're already into some ontology, or you already use it, then it is definitely a big bonus. All right. Again, I'd like to go into a small poll here. Which of these do you think is a part of a biocurator's job? Giving this poll is going to just help me understand whether you have understood everything that is required in a biocurator's job. All right. Now, where can biocuration be applied in research and medicine? Biocuration actually supports a lot of systems integral to result oriented. Okay, so it can give you help pharma companies figure out drug discovery. So if you have a chemical database, they can actually check chemistry and therapeutic benefits of a chemical compound. So that is where they will use it for drug discovery. Precision medicine is which type of cancer has used a targeted approach, has used a cytotoxic drug. So precision medicine, it is going to help you with that, and a lot of other variant interpretation, system biology. So there are a lot of fields in life sciences where biocuration can support or aid further research or result oriented goals. Now, there is a difference in biocuration in the industry and in the academia. So when you are applying, or if and when you are interested and you want to explore this field, industry is a place which is going to focus on a profit-driven project. So it is going to be funded, it is going to be paid for by a client. It is going to have strict timelines, it is going to have strict quality controls, and it is going to be product oriented outcomes. So when it is an industry based approach, or if you're applying to an industry, the industry will tell you, or the client will tell you, that this is what they want. So this is the result that they're looking at, these are the fields that they're looking at, and this is what needs to be curated in so much time. Okay. When it is an academia, it is slightly more flexible. There is more open communication. The knowledge oriented outcome is given more weightage. So it is more about discovering something new or finding something which is missing versus giving a result for a particular block. Okay. So the difference in industry and academia The reason I'm explaining it to you is which one do you associate with? If you ever want to get into biocuration, which one do you closely associate with? If you are in one or the other, it is easier and it'll make you happier if you go into that section. Okay. Now, career paths in biocuration. As Manva has already explained, my career path, I started off as a biocurator, as a very simple editor, in fact, at the age of 24, 25, and have come to a level where I have gotten into corporate development, where I've done various roles in corporate development. So this journey has been a journey of self-learning. This journey has been a learning journey in which I let myself and I let the company to an extent decide certain roles for me, and I kept molding. All right. So it was never a strict thing that I want to do just this or I want to do just that, or give me only genes database or give me only a disease database. So my advice to all of you will always be that stay open. Biocuration is a vast field. People are going to require, maybe today they require a gene database, tomorrow they will require a disease database, a few days later they might expect you to integrate both these together. Okay. So be open to it, try to understand their approach. It is going to help you grow in your wherever you are. I went step by step from a junior role to a very senior role. At the same time, I have been able to balance family life. Just to give you an idea, I have two daughters, so it is always a balancing game. Whether how you do this, how you who do you give more importance to, and when do you give that importance to. So the good part about biocuration is that a lot of it is online. A lot of it is database oriented, so it is on the computer. So you can access some of this if it is your company policy and they allow you. You can access some of these by sitting at home. All right. So it does help you strike a balance. It does help you move ahead, even though your life is changing. All right. The employers, I think there are various types of employers here. The basic broad categories that I've mentioned is research institutes and academia. So you can always choose between these two types and pick your interest. I think we will be providing a list later on, uh, which also encompasses a lot of companies to which you can actually go through and see what each one of them is doing. So with this, I'd like to end my part of the conversation, and thank you. I hope what I've said inspires you, and I'd like to open it out to a small question and answer round, where we can take up a few questions. So yes, go ahead.
Yeah. Thank you so much, Yutika. That was a very nice overview of what biocuration is and what kind of skills are needed, and also what kind of roles can one expect if one wishes to enter this part. And I really appreciate you talking about the last point, which is very relevant, especially as women in STEM. We really need to think about how we can balance everything out when it comes to our balancing the family and the career. So thank you so much. So I would request Vaishnavi to, yeah, let people, give them permission to unmute if they want. If anybody wants to ask questions, just raise your hands or just type in chat or just simply start talking. We are going to have some time for question answers at the end also, but if you want, you can just ask. Akash, do you want to ask something? Yeah.
Yes, ma'am. Actually, I have a question related to gene therapy and biocuration. Does this
Okay.
biocuration helps in gene therapy related issues? Suppose there is a mutation. So, is there any data from like a human genome project we could use to find a new solution or a cure, something like that?
Yes, so if you remember, I had enlisted a certain amount of databases in the presentation. Some of these databases do cover a variant database, wherein you will see that there is a particular gene, then there is a particular variant, and what is the effect of that variant. Okay, so it will give you a guideline, but a treatment per se is something that is to be discussed always with somebody like a doctor or something, to move ahead. So definitely the research part of it can be aided if that is something you're looking at. If you want to aid a research, definitely in gene therapy, this is useful. But an outcome in terms of a treatment, it is something that if there are treatments which are given before, those can be listed. But if it is a new treatment, it is something you will have to consult somebody with.
Thank you so much, ma'am.
Thank you.
Yeah. Shrutika, go ahead, Shrutika.
Hi, very nice presentation, Nitika. Am I audible?
Yes.
Yes.
Oh, it was very informative and insightful. So I wanted to ask, I have done my bachelor's, master's from IISER Pune, and currently I have a neurobiology background. So I was just wondering how, I understand a lot of molecular, from your presentation, you could understand molecular side of it. But as a neuroscientist, a behavioral neuroscience scientist, how can I look at biocuration, or what job roles can I perceive, or what are the avenues?
Yeah. So, what happens with biocuration, like I've told you before, it is not a limited field. So, it is not going to be only limited to genes and variants and diseases. It is also sometimes people do want to know. So, like you said, there are people who are reporting behavioral outcomes in psychology. There is a huge field in which this is also getting reported. There are some companies who are also interested in neurobiological diseases and how people are getting treated with it, and what are the clinical trials currently that are in publications. So every researcher, every country has to give a certain amount of clinical data on a government website. So all countries have this. So extracting this information from them is something in the neurobiology field, is also one database. Okay, so people are interested in this information as well. So definitely, if you approach the right kind of people who are into this, you can get an opening into that.
Oh, very interesting. So you mean, like currently I'm placed in an academic setting. So I saw that academic versus industrial. So currently I'm associated with IIT Kanpur. So you're saying every national import, like, or any institute for that matter, there are job roles that could, like, whom I can approach to for this. I was
Yes, so
just wondering, like,
what I said is every clinical trial that gets published in
I
India
see. Yeah.
is published on a
Okay.
government website. So, for example, the American website is clinicaltrial.gov. So, if you go there, there is going to be a section of just neurobiological diseases. Okay, so you can screen through that, and there are companies who require just that information. There are companies who are into oncology. There are companies who are into pain management. So there are different sections, and if you do find out if there is a company in that field, you can definitely go ahead with that.
Got it. Yeah. Thank you. Thank you so much for the.
We'll take one last question from Bharti, and then we'll move on to Apurva's presentation. Yeah. Bharti, go ahead.
Yeah, hello. Good evening, everyone. Ma'am, actually, my doubt is about like I am a bit aware of this term biocuration. Even I am pursuing this biocuration as my part of my project. I have done my master's dissertation also in the biocuration part only. But the problem is that being a researcher, I'm not able to get a particular kind of opportunity to work being a biocurator. Like wherever I got an link or something about an opportunity, uh, but I'm not able to put my skill set such that it can have a eye on from the HR side. So what are the what is there any scope of improvement to have a particular field job in biocuration, ma'am?
So field job, you were looking for a field job, or are
No,
you?
I mean, basically, trying to say that in this particular
In
field,
this particular field.
yes, like
So,
dry lab based only, ma'am.
yeah, so most definitely we will be sharing a list of companies who are into biocuration and who have been, who hire people from biocuration side. So that will be shared towards the end of the presentation. So I'd request you to wait for that and check those companies and see where your interest overlaps and then apply in one of those, because it is definitely, if you are already trained into it and you've already done it, my suggestion would be try to gain some experience and then try to put that experience up. For example, when I applied to my first company, I applied as an observer for maybe 2 or 3 weeks. I was just observing what people do, and they allowed me to do it because they were not paying me for it. Okay, so try to reroute your ways of approach, and definitely it will help you succeed in what you want to do ahead.
Okay, thank you so much, ma'am.
Thank you.
Thanks, Yutika. There's one question, but I think
Yeah.
you already answered that in your talk. That is it a prerequisite to learn programming languages like R and Python in order to enter a biocurator job?
Uh, no, like it's like I mentioned, it's like a add-on that you can already that you can have. In fact, if you have that, it's very good. So you can overlap with the bioinformatics team in designing the databases also. But it is not a prerequisite. It is not something that is essential.
Okay, thank you so much.
Yeah.
We'll take questions again at the end, when both of them can together answer some questions. But right now, I think we'll move on to Apurva's talk.
Sure.
Yes. Okay. Apurva, are you there?
Hi. Hello.
Vaishnavi, I think you have to make Apurva the co-host. Yeah. Or are you already a co-host?
Apurva is already a co-host.
Ha, great. Okay, excellent. Apurva, go ahead.
Can you hear me? Can you see me?
Yes.
Perfect. Perfect. Thank you so much, Manava, for giving us this opportunity. I think this is a very fantastic platform where industry and academia can collaborate and share their strengths and gain from each other's knowledge. And as Manava, as Yutika has very nicely explained the depth of biocuration, what biocuration is, what are the applications of this. In my presentation, what I will now do is I will give you a holistic overview of what biocuration is, what it is not, and how it is typically done. So, different companies perform biocuration differently, of course, depending upon their requirements. However, there are few set in stone principles or ideologies which typically people adhere to. We will walk through that, and then I will also talk about what next. So biocuration is one aspect, but once you have honed the skills of biocuration, how it fits in a broader industry. The questions which some of you asked, especially Bharti, the question which you asked, where as a neuroscientist, how you can apply the skills of biocuration. So I will cover that aspect, and my journey, like Yutika also covered her journey. Okay, so let me present my screen. Just let me know when my screen is up.
Yes.
Can you see my screen?
Yes.
Yes, yes.
Right. So before we go ahead, this is the first poll question. Now that Yutika has very nicely explained you what biocuration is, I'm sure you will now able to answer what it is not. So can we
Yeah.
have a poll for this?
Yes, yes. The poll is already on. So I encourage people to click the Menti link. I'll paste it here again, and you can, try and answer. I can see that some of you are answering. So whenever we give all of the above option, most people choose to click that. But yes, it's nice. Yeah, people are answering. So it's nice to see that people are engaging in this. Yeah, go ahead, Apurva. The poll will be on so they can answer, throughout your talk. Yeah.
Okay. So now let us quickly go over the holistically over what curation is and how it is typically done. So if I am to summarize what Yutika also covered and what curation is at a high level, basically it is to convert your raw biological data into a decision grade or accountable set of knowledge. Okay. So, and why it is done? Again, the reason being the raw data, a lot of you are working in labs, a lot of you are translational researchers. You know that the data comes with a lot of noise, right? So that data has to be cleaned, that has to be processed, and then based upon the cleaning and the analysis, you can then come to some answers, right? So this entire process is curation, and this is the whole reason why we need curation, so that we are able to use the data we have generated. So this is the why behind this entire massive industry. Okay. So what it typically is, at a high level, it is an act of judgment. You already mentioned now that, it is not copy paste. It is not just tagging the fields in Excel. So as a scientist, as a biocurator, what it is an act of judgment. The biocurator should be able to accurately and precisely identify what is exactly the content relevant from the literature that is going to help me get a decision out of this, come to a decision point. It also involves evaluating the evidences. As a biocurator, you should be able to understand the quality of the evidence, the confidence in the evidence, whether a following piece of evidence is coming from 2 patient data or if it is coming from 100 patient data. Relevance and that contextualization. Sometimes, let's just take for one example. There is a study in which there are only 2 patients which showed a response. As a scientist, I might say that no, this is not a relevant piece of information. But if it is coming from a rare disease which occurs just in one patient in say billion patients, in just one patient shows this particular prognosis, then if I have 2 patients, then probably that becomes a very relevant and contextual data. So your data just cannot be seen in a single dimension. It has to be seen in the dimension of context, it has to be seen in the dimension of quality, confidence, and then across time and space as well. One typical example that I have is couple of years ago, but there is one stage of brain tumor which is called as glioblastoma. So this particular stage is a very aggressive stage of brain cancers. Okay, till 2 years ago in the industry, that this disease term was very typically called as glioblastoma across the entire medical fraternity. But as the research evolves, what people identified is that this stage mimics very closely to a different type of disease called as astrocytoma. So as the research evolves, as there are new methodologies of testing, of sequencing, what people realize is glioblastoma has a subset which has a mutation of IDH gene. So then, with this new information, which was very high confidence data, highly accurate data, which came from literature, WHO recommended that glioblastoma is now renamed as IDH mutated astrocytoma. So this is what I mean by standardization across time and space. Something which was called something else at a previous time point is now changed because of advent of literature. So this all is because of biocuration, that you're able to track this growth, be able to assess how it was qualified previously, how it is qualified now, and then based on all these parameters, you're able to take actionable decisions. You're able to make confident decisions based on this. So this is the what behind the why. Okay. And then once we know why do we biocurate, what it is exactly that we do, then the next typical or obvious question comes is how it is done. Let me take your one example of as a biocurator, I want to curate a clinical database for Alzheimer's disease. Okay, this is a hypothetical scenario that I want to curate a database for Alzheimer's disease. The first step that I would do is I will evaluate what all sources I can use for this biocuration, right? Do I use WHO? Do I use any practice guidelines? Or do I just go to PubMed and gather a lot of different published articles? So this is my first step where I evaluate. Okay. Now, once I, for example, narrow down on WHO guidelines, okay, then the next aspect here is to define from the WHO guidelines what exactly I'm going to extract. So this aspect here aligns with what my customer wants, which industry I'm working on, what is the goal of my company. So this is where you extract your evidence and it aligns with the company goals, with the industry norms, with what exactly the patient wants, what is the gap in the medical field now, which I can address by creating this database. Okay. Now, once that has been identified, next, like Yutika mentioned, there needs to be done some harmonization studies where, for example, a disease is called breast cancer in one paper versus carcinoma of breast in another paper. And when I have 10,000s of papers, I cannot manually track and map it, cancer. This is called breast cancer in one paper. It's called carcinoma of breast in another paper. So there are well-established ontologies or well-established vocabularies which are recognized throughout, and those ontologies can be used to harmonize this data so that the data quality is consistent and it is of a good grade. Okay. So once the data is harmonized, the extracted data, it is then evaluated. Okay. Let me take one example here. Okay. Like I mentioned previously, there are only 2 patients which showed positive response to say for one particular drug. But those 2 patients come from a very rare disease subtype. So in such situation, I have to evaluate, provide weightage to that data, and then decide the confidence of that data. Whether that data should be used directly for recommending some drug to a patient, whether that data should be used to do additional testing, or that data needs to be just discarded because it is not a high quality data, or there are some biases involved in the testing. All of that evidence evaluation needs to be done. And once all of this is done, once you have your data curated, that is when your quality checks come into picture. Because when you are dealing with huge amounts of data, be it manual, be it automated, there can be biases, there can be manual errors. So quality checks and quality assurances is a very important step. So there are different ways of performing quality checks. There are different testing models available in the field. People do manual testing as well. So all of this should be conducted. And once all of that is done, your data, a clean, verified, validated, uh, confident data is ready for versioning up and releasing. So this is a typical workflow of how curation process goes on. Now, one thing, what I typically see is a lot of people interchangeably using the terms curation and annotator, or curator and annotator. So before we move further into other applications of biocuration, I want to just take a couple of minutes and clarify this. These are 2 distinct but very complementary processes. Okay, curation is the what part of the entire spectrum. So what kind of knowledge you want, what quality you want, what structure you want, that all answers the curation aspect. Annotation is a subset within the umbrella of curation where we answer what exactly that means. So as a curator, when I identify what knowledge I want to curate, I need to also know what it means. Taking the example I mentioned previously, when I see 2 patients showing positive response, I need to know what it means in the broader spectrum, whether that is a good data, whether that is not a good data. So that is where your annotation comes into picture. What exactly it contains, whether the variant or a genomic alteration, for example, which you are curating, whether it contains any consequence, whether it is giving any uh, disease outcome. So that will be a part of your annotation. So annotation will answer all these questions. Okay. So this is a subtle but very important difference between curation and annotation. So good curator will always be a good annotator. So far we covered all the aspects of curation. We went into the depths of biocuration. We evaluated different parameters of this. Now coming to a broader picture, the infinity loop of research to application and back to research, where exactly, at what all places is biocuration important, and what all different industries are relevant, and with your skills of biocuration, with your skills of domain expertise where all you can venture. So let me start with the fundamental research. So please focus on this first box of fundamental research. So you all know what is fundamental research. It is curiosity driven research. It uncovers systems, functions. You ask questions, you generate hypotheses, and you come to logical conclusion. You devise mechanisms out of it. So the industries which are involved in fundamental research are academia, research, biotech R&D, sometimes pharma discovery as well, so on and so forth. Okay. Now, based on this basic research, this information is fed into functional validation industries. So these are the industries which are sometimes also the translation labs which you see in research institutes, or also R&D units of a pharma company. So this is the small pivoting point where there is a major overlap. What exactly is done when you are functionally validating your research is you validate your hypothesis. For example, there are 10 papers in the field where somebody has addressed a mechanism of a protein. Now a company comes into picture. That company functionally validates all the 10 papers and says that yes, all the 10 papers are correctly answering this mechanism, and then this mechanism is now validated, tested across n number of samples, no biases, uh, relevant controls are placed, and then it goes to a phase 0 trial. So this is where your function is validated, your fundamental research is validated. Once that is done, when you are moving from bench to bedside, the third obvious step for any data to process is your clinical design. Now, once you have functionally validated hypothesis, functionally validated data set, then your data moves into a clinical direction. So this is a very crucial step which decides which data is relevant to move to medicine or relevant to move to application phase. So this is in this stage that there are a lot of clinically designed trials. There are endpoints of the trials which are determined. And as a biocurator who knows the domain, who knows their expertise when they have done the PhDs or when they have done their masters, the key industries which are open for this particular stage are clinical strategy teams. So a company which is running a clinical trial, they will have a strategy team where the role of the team is to devise strategy, devise the trial design, remove all the biases, establish key endpoints. Okay. Phase 0 trials are also conducted in this stage. There is clinical data management if you're looking for remote or more of a computer based role. There are a lot of data management opportunities, and then there are diagnostic companies who are tracking the clinical designing from stage 0. Now, once a data has been clinically designed, it goes to clinical research. Clinical research, I'm sure you are all aware. This is where your hypothesis is tested on patient populations. So, the phase 1 to phase 3 of the clinical trials is what happens in this particular stage. So, if you are to enter into this stage of data, then there are a lot of contract research organizations which do these studies, and then there are a lot of hospitals who focus on this. Nowadays, there are a lot of companies which are coming up, which perform data management, or they analyze the data, which tons of data which comes out of clinical research, and then they make it ready for submissions, ready for FDA approvals or any funding body approvals. So this is that critical stage of data. And then once the trial is conducted, then there is regulatory trial regulations and rolling out the trial where it becomes enrolling for different populations. It's typically a phase 4 stage of your clinical trial. So as a curator or as a domain expertise, if you would want to work on this particular section of the industry, you can enter the quality teams, you can enter the regulatory teams. So quality teams are where you will be doing QA, QC of the data. And trust me, it's a huge data, multiple GBs of data coming from a lot of patient data, and then lot of noise it has, lot of ambiguity it has. So as a quality team, one has to ensure the data accuracy, the quality standards are met, quality thresholds are met, and then the data is ready for regulatory submissions. maybe for FDA submissions, maybe for ISO submissions. So this is the field where the data is converted into clinical ready phase. And then we move to clinical stage where there is this structural bridge between whatever has been identified is now ready to move into the market. So this is where fields like companies like decision support system, where they help the doctors to identify the data and present it to the patients, or they help the users to know what kind of data is to be consumed. All of that happens at this stage. And ultimately the last stage is where there is point of care application, where the drug is out in the market, or any product, uh, any new regime is out in the market and people are able to use it. So the key industries, if you want to work at the end point of this loop, is diagnostic labs, where you are in touch with the users, or digital health providers, or you can be a clinician associated roles, where you are be a genetic counselor, or you're helping somebody with further growth of the usage of the products. And then from here the data again feeds into the fundamental research, where if required, whatever gaps are there in the research, or whatever newer applications or newer usage evolves, it goes back to the fundamental research. So this is typically how the infinity loop of data moves from research to application. And as a biocurator, or as a person with the biocuration skills and capability and the subject matter expertise, these are all the arenas where biocuration can be used, and your technical skills can also be explored further. Okay. So these are on the same loop. These are some of the transferable skills and relevant roles which you can target. For the fundamental research, you all are aware that the designations, typical designations, are research scientist, investigator, discovery scientist. And what you do at this stage is hypothesis formulation, experimental design, analytical thinking, so on and so forth, right? Now, when it moves to functional validation, the designations here are preclinical scientist, or a systems biologist, or a validation lead. Okay, and what you need at this stage is causal thinking. So in fundamental research, it is more about analytical thinking, but the validation, or the correlation and causational association, that has to be established at this stage. So that's an important aspect to be uh, associated with this particular role. And then interpretations and cross-disciplinary collaborations, that is important for this particular set of roles. And then for clinical design, you would require to know the endpoint definitions. You would be designing the different ways the trials are designed, identifying the biases. So the roles here would be product subject matter expert, design specialist, trial lead, so on and so forth. Now for clinical research, the designations would be research scientist, medical scientist, program managers, and the transferable skills you would require is evidence evaluation, executing the protocols, getting the patient data, updating the patient data, data interpretations. Whereas when you come to regulatory and approvals, this is a huge and independent domain in itself. Regulatory affairs managers, or quality lead, compliance specialist. There is this new industrial jargon, which is SAMD SME. So it is software as a medical device and subject matter expertise for that particular role. And then when you move to clinical readiness and point of care, it will use a combination of all these skill sets, where you would work as a product manager, you can work as a knowledge base lead, clinical informations lead, and also as an analyst or as a real world data scientist. So these are all the key examples of the different industries, different designations, and what skills, in addition to your existing qualifications, you would require to move into this entire spectrum of the industry. So all this is good, this is all a rosy picture, but always there are going to be challenges when you're dealing with huge amounts of data. So some of the major challenges which industry faces today is data harmonization. Sometimes the content is written in a very different way. So like Yutika covered this example, there are different spellings. There are different ways disease is called out. Sometimes different continents call a disease separately. There is differences in which WHO mentions a particular disease versus your literature from a different continent mentions a disease. And there has to be some standardization. So standardizing this and aligning with what your customer wants as an industry person, that becomes a challenge. And same is with the ontologies as well, where your the structuring and the relationships of the data is very critical, and that is sometimes also confusing to the users. What I mean by structuring and relationships is that let me take one example here, okay? Carcinoma of breast is one of the disease subtypes. In one of the ontologies, which is a very well known ontology in the field, carcinoma of breast falls under solid tumor. So, if I am to structure my data, I have solid tumor as the most important or a bigger branch of diseases, umbrella of diseases. And within that solid tumor, one of the subtypes would be breast cancer. So this is the way this particular disease is structured. Now, if I go to a different ontology used by some different companies or used by different physicians in the world, breast cancer would fall under BRCA mutated tumors. So this is a different, it's the same disease. The disease will say show same symptoms, same prognosis, same therapy, but the way it is structured changes. Now imagine a huge company running a clinical trial, which clinical trial of say 10,000 patients, which is trying to establish this relationship. It becomes difficult. If you're dealing with 10 patients, you can simply do it manually, and you can apply the logic, you can apply your expertise, knowledge expertise, and say, oh no, this is the same disease. If you're doing it for 10,000 patients and for 10 different trials, that becomes a huge thing, and that becomes confusing as well. So this is one of the major challenges we face currently. Third challenge is regulatory readiness. Adhering to ISO regulations, adhering to FDA norms is a very nice way to ensure your data quality. It gives you an edge when you are an industry person, that your data is of a good quality. However, that there is always a very fine balance when you are trying to adhere to regulatory parameters and trying to deliver your data to the clients on time, because you always know, right, that the curations you are doing is going to be used by some patient or is going to be used by somebody in the industry who will either develop a new drug over it, who will either use that data for something. So moving that data from research to decision making point has to be quick so that it can be used very soon. But along with that, there is this trade-off of ensuring your data is of good quality, data is compliant, data is traceable. So this trade-off or this balancing is a very fine and very critical activity that has to be managed. And one last or another big key challenge is evidence conflicts. Different research papers, a lot of you would have observed this. When you identify a research paper where they have listed some example or some result, you might find a paper which says exactly opposite of what that paper says. Does that mean one of the paper is wrong? We don't know, not necessarily. Sometimes the situations or the backgrounds are different. One drug which is showing a response in 10 patients might show zero response in another 10 sets of patients. Reasons could be different. Reasons could be a mutational background is different of that patient, the lifestyle is different, the age is different, the gender is different, sex is different, but there are evidence conflicts when we see. Now, as a biocurator, when you're trying to generalize this data or make this data consistent, this becomes a challenge. So then establishing the methodologies to evaluate this data, to understand the bias in the data, and to come to a logical conclusion, and take both the conflicting data points and then come to a logical end saying that this is the ultimate truth, which will help you in a decision making aspect. That is a very critical and that's a key challenge. So these are some of the key challenges we typically face when we are when we are biocurating or when we are dealing with data which is being used for any of the downstream activities. Okay. With this, basically, the next step, what I want to talk about is key differences between industry and research mindsets. Okay. both the industries, both the industry as well as research are very unique in themselves. There are very crucial skills which one needs in each of these aspects. So there are certain overlaps, but there are certain contrasting aspects as well. And personally, when I moved from research to industry, I had a tough time wrapping my head around it and identifying why is this not done. I mean, I came with a very fantastic idea, and then why somebody in my team is not agreeing to it. But then when you look at it from an industry standpoint, you realize that your curiosity has to be boxed in with certain norms. Okay, so let's see what that is. So when you are in a research industry, it is always curiosity led exploration, right? You are working in particular lab, you know what question you are trying to address. You don't have an end in mind, and you should not have an end in mind. You should be doing the research, and whatever result you see, that is going to be your result, be it positive result, be it a negative result. However, when you are in an industry, your curiosity is guided by very clear goal. When you are working, for example, in a say a digital platform, the goal will be to provide a relevant drug to the patients. Whatever is a relevant mutation that patient has, the relevant therapy for that mutation has to be presented. If that is my goal, I have to read the research papers with that goal in mind. I have to read research papers of that domain. For example, if I am working on patients, Alzheimer's patients, if I'm working on creating database for Alzheimer's, I must read Alzheimer papers. I cannot just, if I have curiosity for diabetes, and I cannot just sit and read diabetes papers. I have to be goal driven, right? When I'm in a research mindset, I will talk about what is true. But when I'm in an industry mindset, I will think about what is useful and what is scalable now. The research that I'm doing can be used for next set of researchers, but in an industry, the role I am playing or the work I am doing has to be useful for some scalable outcomes. There is a knowledge generation when we are into research, but there is impact adoption and outcomes when it comes to industry. And in research, there is always novel insights or discovery, but in industry, there always has to be a trade-off between rigor, speed, and value. Research is long term, exploratory, but industry is time bound. It is milestone driven. So these are the key aspects I would want you to keep in mind, because that helps to attune to what you are uh, looking for in the industry, and then help you align your expectations according to that. With that, I will quickly walk you through my journey. The reason I want to walk you through this is because at different time points, when I played different roles, there was a switch in the skill sets which I had. And trust me, these are all adaptable skill sets. Nobody's born with this. You can always unlearn these skill sets, and you can move to the next roles. So initially, when I was a research fellow at ICER, like you all, I was working on hypothesis, methodologies, publications, mechanistic testing. Then, as a genomic scientist in a diagnostic lab, my role was to perform experiments with a very explicit quality parameters, very well-defined output points, and ensuring that there are outputs that I get in a very timely manner. Okay. And then I when moved to curation as a biocurator or a senior biocurator. My role was to curate the genomic content for oncology, and also as a senior biocurator, my role was to review the content, do the sample testing, do regression testing, perform verification, validation of the data sets, ensure how I'm able to automate the data. Okay. And then as you move into a leadership role, your skill sets or your thought process pivots a little bit, and you now start thinking at a systemic level where you are ensuring the data which is going out from your team is good quality, or you're establishing curation approaches or strategies at a company level. Okay. And then as a product operations manager or a product manager, this was again a pivot from pure curation or pure scientific work to more of a applied work, where I was working at a edge of curation, science, strategy, and business. So here the role is to manage operations of different products, where curation is one component, software is the second component, business is another component. So if you know your domain, if you have the subject matter expertise, then adapting these other skill sets becomes easy because you know the content, because you know your industry. So this is the second type of activities where you perform as a product manager. And then you also build healthcare products. You are involved in working for decision making at a company level, or you are defining the business requirements. So these are the different skill sets you require for different roles which you perform. This is my short journey. And if you would like any further help from me, I am open. I have pasted my QR code here. This is my LinkedIn QR code and the email. If you want, you may contact me. And if you have any questions, I'm happy to take those as well. Thank you.
Thank you so much, Apurva. Thank you. I will share both of your LinkedIn profiles as well as email addresses if you're okay with it on the webinar WhatsApp group also. But that was a very nice overview of your journey, and it sort of helped us see that one always has to keep on transitioning. And Yutika also said that just be open to learning and be open to, whatever comes your way. So that's a very nice approach, which is what I could see is common in both of your journeys, that you did not stick like this is what I want to do, or this is the field that I want to work in, this is the role that I want to do. Because eventually one has to shift from basic research, one has to develop an industry mindset also, and also go into people management eventually, which is something that we are not trained for at all during our PhD. But we learn on the job, right? So there are a couple of questions. We'll address those. Either of you can take this, and then we'll go for specific questions if people want to ask Apurva or Yutika something. Somebody was asking if such kind of roles are available for in material sciences. Do either of you know about this? Because there are a couple of, because this was open for all STEM PhDs and STEM ECRs. So
I think I have come across one project which was a very small ad hoc project which we had done, where they had used not us specifically, but the people were using somebody with this background for designing drug delivery systems, wherein they wanted to standardize the porosity, the composition, the material descriptors, and things like that. So that is one thing that I have come across once before. But other than that, I have not really come across anything specific. But this definitely was a role where that person was from the material science and was to standardize material science and biological descriptors together and then
okay.
make a drug delivery base.
Okay, interesting. So it was still in the context of healthcare and yeah.
Yeah.
Okay, nice. There's another question which maybe Apurva can take. Is there an on-job training provided by the companies who employ biocurators?
Yes, absolutely. There is on-job training because different companies will require different kinds of biocuration from different sources, which is relevant for their products, for their clients. So there is always on-job training. Yes.
Okay, thank you so much. If there are any other questions, you can simply maybe now unmute and you can unmute and ask questions. You can raise your hands. There is somebody asking in the chat, is there any app for biocuration?
App, not that I've heard of. There is, however, a LinkedIn group where a lot of different biocurators connect. Yeah, I think that should help you.
Okay. You can just simply raise your hands and ask us questions, or you can type your questions in the chat or in the WhatsApp group. As Yutika mentioned, so we have a list of companies which are, working in these fields. So if people want, they can reach out to me and I can share this information. I'll also share all of this information on the WhatsApp group. Yeah. And yes. Naina Pawar ma'am is asking, can we apply it in medicinal field?
Yes, in medicinal field as well, there will be a lot of biocuration opportunities involved. and it will have different aspects, biocuration, clinical trial data management, a lot of different aspects. So yes, you can do apply.
Yes, especially if you're from the pharmaceutical chemistry side, there are a lot of drug databases which deal with uh, where they want chemical structures and their therapeutic benefits. So it will definitely can be applied in the medicinal field.
Okay, thank you. Dr. Kash Kumar, please go ahead and ask your questions.
Hello, am I audible?
Yes, please
Okay.
go ahead, sir. Yeah.
So primarily, I'm thankful to iRISE and both the speakers for such an enlightening talk. And the question is related to like data we usually submit on these databases like NCBI or Ensemble. So if you are submitting a data on let's say NCBI, let's say RNA-seq data or some genomic data, the data will go to some biocurator at NCBI or how the process is, and let's say we are not getting any query on our data after submission, it means all the curation done by us is completely correct, done perfectly, or they will do something on that data later on as well.
Manoj, Yutika, you want to take it?
I think you can go ahead. So it is, according to me, it is not like the end of a submission. There is no end usually to it, but it is always reprocessed and it is then updated and then it comes back to you. So it is validated at some time point and then it will come back to you again. So is that what you're asking? Did I
No,
answer your question on that?
No, actually, whenever we submit a data, for example, genomic data, RNA-seq data on, let's say, NCBI, so there is a complete procedure of multiple steps, and then you have to upload your data through some links or high-profile internets and all. So, I mean, it's a time-taking process. In fact, author has to put a lot of effort in that while uploading the data on that particular platform by filling a lot of forms, and then steps are there to be followed precisely. If you are not following that steps, they will definitely reach back to you. In fact, next step will not be proceeded as such. In my case, I have submitted RNA-seq data and one genome data as well. But after submission, I've only received a submission email and a date in which data was published online. But no, I mean, they didn't raise any query as such. So my question is, whenever we are submitting such data to the NCBI, are there biocurators? Those are actually checking our data.
So, yeah, that's a good question. Yes, there is biocuration involved at this stage. Now, depending on where you have submitted and their processes, there could be some automations or AI at an initial stages where they will annotate your data, they will assign relevant tags to it, process it, and add it to their databases. At a further time point, there could be some manual interventions and manual biocuration involved. But if you have not heard back from them, which means there is no doubt, and probably the data has been annotated and is in the database now.
Okay.
Okay. Thank you.
Yes. Akash, there's a question. There are a couple of questions in the chat. We'll take those and then Akash's question will be the last question because we are running out of time. Yeah. So Quali is asking, hi Quali. Quali is asking, being completely in academics teaching, how can I train myself for biocuration and molecular biology? Is there any online course or hands-on workshop? There is a question whether there's an application in plant science. And again, Shrutika was asking about the role of AI, which Apurva also mentioned briefly right now. So these are the questions. Yeah. Then we'll end with Akash's question. Yeah.
So,
Sure.
yeah. So, Dr. Kwali, there have been many people who have come from the academic background who have also evolved themselves into biocuration. Currently, there are a few online workshops which you can access. Apurva, if you know the details of those, do add in. But in general, like I think once mentioned before, that the company that you apply to will usually give you a hands-on training, because when a company takes up biocuration, it is very specific to that company. So they will definitely invest some time and training into making sure that you're curating as per their standard guidelines. So that part will probably be, managed a little bit later by your company. But if you do want to do anything online, Apurva, are you aware of any online programs which
There are one or two courses on Coursera and Udemy which you can look at. That will definitely help. Apart from that, if you're coming from academia background or teaching background, a lot of transferable skills which you already have will be applicable, right? Like attention to detail, which is it's a very cliche thing to say, but it is a most important skill when you are a biocurator. Attention to detail, identifying the data accurately, and making sense out of it, looking at that data in a bigger picture, not just getting one data point from a paper, but holistically understanding the paper. So with these basic foundational skills and with one or two courses, and then in a company when you go, you will get trained. That should be a good substantial training to be a successful biocurator.
Great.
Thank you,
Thank
ma'am.
you. Yeah. Thank you. Akash, do you want to ask your question?
Yes, ma'am.
Yes. Yes, go ahead.
Go ahead.
Ma'am, actually, as you are experts related to biocuration and all, and very close to industry, so are there any roles or like startup wanted by the industry so that they can bridge the gap between biocuration and application part? Hello.
Hello. Yes, I can hear you. Could you please repeat your question?
Are there any roles which industry is seeking and there is a gap because you guys know industry and
I'm sorry, Akash, you're not audible. Could you just type the question? If I understood his question correctly,
biocuration.
Akash, I think you're
So
not
much.
audible. Just type the question. Yeah, I think we can't hear him, but I hope he will type his question. But I think he was talking about bridging the gap between maybe industry and academia. And that is a question which we keep on getting at our ECR workshops. That, here also in the chat, we are getting questions like, are there any specific courses? Are there specific workshops that we can do to actually enter the industry? So we got the point that once we enter the industry, we will get on-job training. But the key point is entering the industry. And that is where we need to highlight the skills that we develop in during our PhD. As Apurva had mentioned in her slide, they are transferable skills. So that is the problem which a lot of PhDs face, that they don't know that they already have a lot of transferable cross-cutting skills. They don't know they have them, and they don't know how to highlight them or showcase them. So to enter the industry, to enter different fields. So that is something that we will definitely focus upon in our upcoming workshops also. But thank you so much, Yutika. Thank you so much, Apurva, for giving us a very nice overview of the biocuration field and what kind of roles could be present for biologists or life science graduates in the industry. Yeah. Okay. I think Akash has typed industry related skill set in the current trend. So that is what he was talking about, bridging the gap. Yeah.
So that is covered in one of my slides and also in Yutika's slides, where we talk about transferable skills at each different time point in the data loop.
Correct.
So you can look at those skill sets, and
Yes.
that should help you basically.
Yes. Correct. Okay. So thank you so much, both of you. I would like to thank our funders again. I would like to thank ICER and our entire team. Thank you, Dipali. Thank you, Vaishnavi, for making sure that the webinar and the streaming is running smoothly. So this was nice. Please, we'll share a feedback form with you. Please enter your give us your honest feedback, and we would also like inputs from you about what should be the topics for our further webinars or one-day workshops, right? So thank you so much everyone for joining on time and attending. We've way past the time, but it was a very fruitful discussion, and I also learned a lot about biocuration. So thank you so much.
Yeah.
Thank you.
Manva, one request
Yes.
from my side.
Okay.
Can everybody switch on the cameras? We'll
Yes.
take a group photo
Yes,
of
I
this webinar.
forgot that. Yes, I forgot that. Thank you, Vaishnavi. Okay. I'll just remove spotlight so that we all can see. Yeah.
We have to take two group photos since I think more than
Yes.
50 people are
Yes.
there. We'll take it in a two
Yes.
sets.
Yes.
Everybody, please switch on the video cameras, please.
Yeah. It's nice to see so many familiar faces.
Yeah, I
So,
was about to say the same thing. Nice to
yes.
see so many familiar faces.
Yeah, because people are, so we circulated information about this mainly in our level 1 workshops, which we do on site. And also I see a lot of
काय
teachers.
कशी आहे तब्येत बरी आहे?
माने सर, तुम्ही आम्हाला विचारताय का तब्येत बरी आहे का?
नाही मनवा मॅडम, एक गेस्ट आलेत आता.
ठीक
प्लीज प्लीज प्लीज.
आहे.
बरे आहात?
हो हो बरे. हो.
हा, हा.
येस. हा.
Okay, so
Okay. Smile please.
first group photo that I'm going to take right now. Just on the count of three. 3, 2, 1. Done.
Thank
Now I'm
you.
going to change my screen. Please switch on your cameras. I'm requesting once again to all of you. Switch on your cameras. I'm still seeing, I'm just seeing the screen only.
Yes. No, now people have started switching their
Yeah.
cameras on. Yes. I'm seeing so many familiar faces. It's so nice.
Okay. Just a few seconds. Okay, I'm going to take another group photo.
Yes.
On the count of 3, 2, 1. Yes, thank you.
Lisa is smiling. I can see that. Nice. Yes. Great. It was lovely having all of you here today, and thank you once again, Yutika and Apurva.
Thank
Ah.
you so much.
Hello,
Thank
It was a
Hina.
you, ma'am.
privilege.
Yes.
Hello, ma'am.
Ma'am, I wanted to ask when there will be round 2, level 2 of the already iRISE, which have been held last year.
Yes, we will let you know. We will definitely let you know.
Thank you, ma'am.
Yeah.
Thank you so
And
much.
it was a wonderful session. Thank
Yeah,
you so
and
much, everyone.
what I wanted to mention is that many of you are teachers who and you have MSc and PhD students in your academic institution. So even if they could not attend right now, do pass on this information. Do talk to them about this because that's a very viable career option which they can now pursue. Right? Shubham, yeah, exactly.
Thank you so much, ma'am.
शुभमला बघूनच आठवण झाली मला याची. हा.
Yes, ma'am.
Ma'am,
ठीक आहे.
please share the
Sure,
web
ma'am.
companies, list of
Yes,
companies.
I'll share. So I'll share if Yutika and Apura are okay with it. I'll also share PDFs of their presentations as well as the list. And do answer the feedback form. Thank you so much.
Thank you, ma'am. Thank you.
Yes,
so much,
thank you.
Thank you, ma'am.
To everyone.
Yeah,
Have a
thank
good
you.
day.
Yeah, you
Bye.
too.
Thank you.
Shrutika, hi.
Thank you, ma'am.
Thank you so much.
Thank you. Yeah, we'll share the feedback form via email also and on the WhatsApp group also. Raghavendra sir. Okay.
हॅलो मॅम, कशा आहात?
मी छान. तू काय म्हणतोस? तू कुठे आहेस
मस्त, ma'am.
आत्ता?
मी केजे सोमयामध्ये आहे.
समय.
Yes,
चांगलं
ma'am.
आहे. सो सध्या MSD बंद आहे त्यामुळे पण मला
हो.
MSD भरपूर लोक दिसले. माने सर होते,
Yes.
गुगे सर होते.
चालेल.
मॅडम
बोला. बोला माने सर.
जरासं जरासं थोडंसं अजून जरा प्रिसाईज आजचं जर सेशन झालं असतं तर बरं झालं असतं.
ओके, येस, करूया आपण.
कारण
हा पहिला, हा,
एवढं आम्हाला नाही हे लक्षात आलं.
बरोबर. नाही, कारण
पण-
हा ओरिएंटेड जास्ती पीएचडी स्टुडंट्स करता होता, प्रिन्सिपली जे आत्ता पीएचडी करतायत बायोलॉजीच्या विषयांमध्ये आणि ज्यांना बायोक्युरेशन मध्ये एंटर करायचंय. पण हे
अच्छा.
चांगलं आहे की इतके शिक्षक पण जॉईन झाले, कारण मग तुम्ही तुमच्या स्टुडंट्सना, म्हणजे तुमचे स्वतःचे पीएचडी स्टुडंट्स असतील किंवा तुमचे रयतच्या इतर कॉलेजेसमधले शिक्षक असतील ज्यांना मे बी रिसर्च किंवा टीचिंगमध्ये नाही जायचंय पण वेगळे करिअर ऑप्शन शोधायचे, तर त्यांच्यासाठी तुम्ही आता त्यांना ही सगळी इन्फॉर्मेशन थोडीफार हे करूयात. पण हे
बरोबर.
तुमचा फीडबॅक आम्ही नक्कीच नोट डाऊन करतो की थोडा अजून स्पेसिफिक करू शकलो असतो आपण. आम्ही करतो म्हणजे आता नेक्स्ट टाइम, कारण आता आम्ही पण हळूहळू विचार करतोय की कसं हे पुढे न्यायचं. सो आता अजून काय काय कुठल्या कुठल्या थीम्स वरती वेबिनार्स करायचे मे बी असं करतोय विचार. येस, थँक्यू. येस, थँक्यू.
वेलकम. ओके.
ओके, या. I think we can close the meeting now.
Yeah, we can close the meeting, right?
Yes, yes. Okay. Thank you.
Thank you.